All right, today we're jumping into a really powerful technique called gas-liquid chromatography. And to get our heads around it, we're going to think of it as a kind of high-stakes, microscopic race, a race for chemicals.
So, let's start with a challenge. Imagine you had to find one single specific drop of a pollutant in an Olympic-sized swimming pool. I mean, how in the world would you even begin to do that? It sounds impossible, right? But this is exactly the kind of problem scientists deal with all the time.
Well, the answer is a technique called gas-liquid chromatography, or GLC for short. It's this incredibly sensitive method that lets us take a complex mess of chemicals, separate them out, identify them, and even measure them. And the absolute best way to picture how it works is to stick with our analogy. It's a chemical race. Let's see how it runs.
The whole process, from start to finish, really breaks down into four main stages. First, you inject the sample. That's the runners getting into the starting gate. Then, they separate as they all race down the track. Next, a detector clocks each one as it crosses the finish line. And finally, we analyze the race results to see who was there, what their finish time was, and how big their team was.
But before we even get to the starting line, let's take one step back and understand the fundamental problem that GLC was invented to solve. Let's talk about the separation challenge.
You see, the whole point here is basically deconstruction. You start with a kind of chemical soup. Maybe it's a water sample or a new perfume or a medicine. It's all mixed together. The goal is to pull out every single individual ingredient from that soup so you can figure out exactly what it is and precisely how much of it is in there.
Okay, so let's follow our chemical runners as they get ready for the race and actually enter the machine.
So it all kicks off at the injection port. The sample gets hit with a blast of heat that instantly turns it into a gas. It's vaporized. At that very same moment, a steady stream of what's called a carrier gas, usually incredibly pure helium, starts pushing that chemical vapor forward. Think of it like a constant wind at the runners' backs, pushing them from the starting line onto the racetrack.
And this brings us to the absolute heart of the entire system, the racetrack itself. In scientific terms, this is called the column.
Now, this racetrack has two key features that make the whole race possible. First, you've got the mobile phase. That's our helium carrier gas. It's always moving, always pushing everything forward. And then, lining the inside walls of the track is the stationary phase. This is a super thin, microscopic liquid coating, and you can just think of this as the sticky part of the track.
So here's the magic. This is how the separation actually happens. Different chemicals interact with that sticky stationary phase in different ways. Some of them aren't very sticky. They have a weak attraction, so they spend most of their time in the mobile gas phase just zooming right along. They're the fast runners. Others, though, are very sticky. They're strongly attracted to that wall coating, so they get held back, slowed down. And it's that very difference in stickiness that separates the entire mixture.
Now, there's a scientific term for this stickiness. It's called the partition coefficient, or Kc. It's really just a number that tells us how much a chemical prefers to hang out on that sticky stationary phase versus being carried along by the gas. So a chemical with a big Kc value, that means it sticks really well, and it's going to be one of the slow ones with a much longer race time.
So after all the runners have separated out along the track, they start to approach the finish line. And in GLC, that finish line is the detector.
As each chemical, now all by its lonesome, finally exits the long racetrack, it flies directly through a detector. You can think of this as a super-sensitive, high-speed camera at the finish line. Its one and only job is to see every single runner that crosses and to send out an electronic signal for each one.
But, not all finish line cameras are the same. Depending on the kind of race you're running and the kind of runners you have, you're going to need a different kind of detector to see them properly.
This chart lays it out perfectly. You have universal detectors, which, as the name suggests, can see pretty much anything. The flame ionization detector, or FID, is a classic example. It's the workhorse of the petroleum industry because it can see nearly any compound with carbon in it. But then you have selective detectors. These are specialists. The electron capture detector, or ECD, for example, is incredibly sensitive but only to specific things, like the halogenated compounds you find in pesticides. So you pick your detector based on exactly what you're trying to find.
Okay, so our detector has done its job. It's generated a whole series of little electronic blips. How do we turn that into something that we can actually read and understand?
Well, those electronic signals are sent to a computer, which turns them into a graph. And that graph has a name. It's called a chromatogram. This is it. This is the official race result. Every single one of those peaks you see represents one specific chemical that has just finished its race and crossed the finish line.
So, looking at all those peaks, how do we know who's who? The key is something called retention time. This is the official finish time for each chemical, measured from the exact moment of injection to the moment it hits the detector. And here's the beautiful part. Under the exact same race conditions, a specific chemical will always have the same retention time. It's like a reliable fingerprint that lets us identify it.
So that chromatogram gives us two absolutely vital pieces of information. First, there's qualitative analysis. That's the who. Who was in our sample? We answer that with the retention time. If a peak shows up at 5.3 minutes, we can match that to a known standard and say, aha, that's caffeine. Second, you get quantitative analysis. That's the how much. And we figure that out from the size, specifically the area, of the peak. A bigger, fatter peak means there was more of that substance in our original sample.
And this is so much more than just a neat lab experiment. This chemical race matters immensely in the real world. We're talking about its use in environmental monitoring to find those tiny traces of pesticides in our water, in food safety to check for contaminants, in forensic science to analyze evidence from a crime scene, and in making sure the medicines we take are pure. It is an absolute cornerstone of modern analytical chemistry.
So, gas-liquid chromatographically gives us this almost magical ability to see the invisible chemical world all around us in incredible detail. And that leaves us with a really fascinating thought. When you have a tool this powerful, a tool that can find almost anything, what are the most important things we should be looking for?